So now we're going to talk about the AstraZeneca-Oxford vaccine. And we're going to compare and contrast to the ones that have gone before it, like the Pfizer-BioNTech vaccine and the Moderna vaccine. Now, as you will recall, the Pfizer-BioNTech vaccine and the Moderna vaccine are basically new vaccines in the sense that they are injecting messenger RNA into your cells to tell the cells to produce the protein that is the same as a portion of the spike protein on the virus, and that is what generates the immune response. So in other words, we're giving instructions to the cell to make the foreign protein which causes the immune response. And that is actually very similar to the AstraZeneca-Oxford vaccine, except instead of using messenger RNA, they are using DNA to do that. So once again, in terms of a picture, imagine we have the cell and we've got the nucleus. And what we were doing before was we were taking messenger RNA, and that messenger RNA was being translated by ribosomes and converted into the protein. And what we did with the Pfizer-BioNTech vaccine and the Moderna vaccine is we were getting the messenger RNA into the cytoplasm of the cell. The cytoplasm is the outside portion, as opposed to the nucleus. How do we get that messenger RNA in the Pfizer-BioNTech vaccine and the Moderna vaccine in? Well, we used a lipid bilayer, or a micelle as we called it, to get those fragments, or that messenger RNA, into the cytoplasm. And then from there, it's fairly straightforward. The cell uses its own cell machinery, which is the ribosomes, to cause translation to occur, and you make the protein in question. How this is different is AstraZeneca uses a different technique, and it uses a virus called an adenovirus, but not a human one, but a chimpanzee. Now, why would it do that? Chimpanzee adenoviruses have not really been exposed to the human population, and so it's very unlikely that humans would create an immune response against the adenovirus specifically itself. We don't want that to happen, because if that happens, then the vaccine is not going to be effective at getting its instructions into the cell. And of course, AstraZeneca knows that if this vaccine is successful and it's used on a number of people, millions, billions of people worldwide, then it's probably going to have to abandon the chimpanzee adenovirus platform for delivering instructions. Now, the adenovirus here does not use RNA like we've been talking about. It uses DNA. And DNA is a double-stranded form of instructions. Now, there's a portion there in the middle of that is used for replication of the virus. Well, the genetic engineers simply delete that portion so the virus can no longer replicate and cause an infection in the human body. And instead, they replace that portion of the DNA with the genetic code, the instructions that the geneticists have engineered. And so what happens when you deliver this adenovirus in the vaccine, it goes into the cell and it releases the DNA into the cytoplasm. And then that gets taken up and goes into the nucleus. That DNA, according to scientists at AstraZeneca, does not get incorporated into the host's genome. It does not go into your DNA in the cell, but rather it is transcribed into RNA. And that RNA then exits the nucleus as a messenger RNA, and then exactly the same thing happens as what we saw with the Pfizer-BioNTech vaccine and the Moderna vaccine, which is that the messenger RNA, which is now a product of the transcription of the DNA, gets converted into the spike protein through translation by the ribosomes, and then this goes on once again to cause the immunogenic response.